Okay, so today we're going to talk a little bit about dopaminergic interactions between commonly abused drugs, specifically cannabinoids and opioids. And to do that, we're going to cover a little bit of the background on why dopamine is important in addiction, some of the mechanisms through which cannabinoids and opioids elicit dopamine release, and potential mechanisms for cross-tolerance between the drug classes. We're going to highlight some key findings from this research study that we published in Dr. Olson's lab in 2020 and then discuss how these findings relate to the abuse liability of the drugs themselves. Now, dopamine has been a focus of a lot of addiction research for more than 50 years, and I'm just going to cover a few of the key elements here. Now, there are multiple dopamine pathways in the brain, but we're going to focus specifically on the mesolimbic pathway, originating in the ventral tegmental area of the midbrain, with its primary projection target, the nucleus accumbens, in the forebrain. And it's really dopamine release in this region specifically that's garnered a lot of attention in the addiction field. Dopamine is released naturally day to day by motivating stimuli such as eating, but drugs like cocaine hijack this process and elicit much, much larger amounts of dopamine in this region. And this can happen by increasing either the frequency of neuronal firing and the frequency of dopamine release events, or the amplitude or the quantity of dopamine that's released during these events. And while dopamine release in the nucleus accumbens isn't the only mechanism of interest, it is regarded overall as one of the first steps in this cascade of neurophysiological events that over time leads to changes in the brain and ultimately behavior, resulting in compulsivity, loss of control, really just uncontrolled drug seeking and drug taking behavior. So a variety of drugs elicit dopamine release in this fashion. These include alcohol, benzodiazepines like Xanax, psychostimulants like cocaine and amphetamine, and the two of interest here, cannabinoids and opioids. And these two drug classes are of particular interest for a few reasons. One of these being that a lot of people with substance use disorders are polydrug users, including numerous people that have opioid use disorder and also use cannabis. Cannabis use itself in the general population, it has been steadily on the rise for decades, and there continues to be more and more potent products available on the market. Now, some people refer to cannabis as a gateway drug, implying that if you already start to use cannabis, you may be more likely to then seek out quote unquote harder drugs like opioids. And this has been heavily debated in the research field for a while, and there's mixed evidence for this. However, one key fact is that statistically, prior cannabis users are about 2.5 more times more likely to transition from opioid use to opioid use disorder should they begin to take opioids. And with opioid related overdoses in the USA reaching over 100,000 annually for a few years in a row now, it's really critical that we understand how different patient histories, including cannabis use, affect the abuse liability if they're to maybe become prescribed an opioid for pain relief later on. And understanding this better will allow us to make better decisions to hopefully avoid these critical overdose situations. Now, one thing we do know is that cannabis exposure does produce cross tolerance to the analgesic or the pain relieving effects of heroin. But what we really want to know is whether cannabis use produced cross tolerance to the ability of heroin or other opioids to evoke dopamine release, thereby affecting their abuse liability. So just to review some of the mechanisms, here we can see a GABAergic neuron terminal in the VTA. And these terminals can come from projection neurons from outside regions or interneurons within the region. These terminals are tonically active, releasing GABA onto VTA dopamine cell bodies or dendrites, where GABA receptors are expressed. This provides a level of tonic inhibition of VTA dopamine neurons, tonically regulating downstream dopamine release in the nucleus accumbens. Now, these terminals can express cannabinoid receptors, opioid receptors or a combination of both. Here is shown the cannabinoid 1 receptor and the mu opioid receptor specifically. And when respective compounds, like THC binding to the CB1 receptor or heroin binding to the mu opioid receptor, they function to affect downstream dopamine release in similar manners. They're binding to these inhibitory G protein coupled receptors, initiating this cascade of G protein signaling, which inhibits vesicle release from the GABA terminal, removing that tonic inhibition and resulting in increases in frequency and potentially amplitude of downstream dopamine release in the nucleus accumbens. Now, there are a few potential mechanisms of how tolerance and cross-tolerance can develop within and between these receptor signaling systems. The first of which involves the G proteins themselves, such as G protein sequestration or G protein decoupling, which effectively limits the ability when the ligands themselves bind to the receptors. It's limited the availability of these G proteins to actually engage in their signaling cascades, thereby reducing the effect that the ligand has on intracellular signaling. The receptors themselves can also become internalized, leaving less receptors available on the cell surface for the ligands to bind to. And there are potential mechanisms by which the receptors themselves can be altered such that the ligands have lower affinity and don't bind as effectively to them. And all of these mechanisms can lead to the same dose of drug having a lower or even no effect on the GABAergic terminals and thus downstream dopamine release, allowing that tonic inhibition to proceed. So to measure these dopamine release events, we use a technique called fast scan cyclic voltammetry, where an electrode, such as the one pictured here, is implanted into the nucleus accumbens of a rodent subject, in this case a rat. And then the voltage of this electrode can be manipulated such that when dopamine is present and comes into contact with the electrode, it will either gain or lose electrons, giving off electrical feedback that we can measure to infer the frequency and amplitude of dopamine release events. So initially, we wanted to just look at how cannabinoids themselves were affecting dopamine release. And we used the synthetic cannabinoid WIN, which is a fairly potent cannabinoid. And when WIN was administered, we did see a significant increase in both the frequency and amplitude of dopamine release events in the nucleus accumbens, showing that the cannabinoids themselves can be effective dopamine releasers. Then we wanted to see what would happen with repeated administration. So following 5 days of repeated administration, we then gave the same dose of our synthetic cannabinoid. And in the blue, you see the typical increase in frequency of dopamine release events given with the synthetic cannabinoid. However, after 5 days of repeated administration, we can see this is significantly reduced, where the administration of the drug is no longer creating as large of an increase in the frequency of these events. In fact, it required about a fourfold higher cumulative dose to produce the same relative increase in frequency of dopamine release events. So this is showing that cannabinoid administration can produce tolerance to its own dopamine releasing effects. The next step was to see whether it would produce cross tolerance to the dopamine releasing effects of heroin. And so we see here in the blue that heroin does dose dependently increase the frequency of dopamine release events in the nucleus accumbens. However, it doesn't have much of an effect on the amplitude. Now, with 5 days repeated administration of the synthetic cannabinoid, we did see that there was a significant reduction in the ability of heroin to evoke increases in frequency of dopamine release events, such that while there is visually a little bit of an increase here in the yellow, this increase, even at the highest dose, was not statistically significantly greater than the baseline. So this is evidence that cannabinoids can produce cross tolerance to to heroin's ability to evoke dopamine release. So what does this ultimately mean? Now, the typical process of taking a drug and becoming tolerant to it has been shown to potentially be a key driving factor in addiction behavior. The drug may become less rewarding or less motivating, and therefore you take more to try to achieve the same rewarding stimulus. This is often referred to in popular literature as chasing the dragon. But what about when tolerance is present from the start? In this case, you have prior use of one drug and then you transition to taking an opioid maybe, and it's already not eliciting as much dopamine release. Now, does this mean that you're less likely to take more of that drug? Or is it still crossing some threshold where it's still rewarding enough, but has a reduced rewarding capacity, and therefore you're still going to end up taking more drug anyway? This also doesn't really take into account the evidence that I mentioned that cannabinoids produce cross tolerance to the pain relieving effects of opioids. So it may be that if someone is prescribed opioids, they already might need to take higher amounts if they're a prior cannabis user just to achieve the same pain relieving effect. And so this may compensate and still produce the same relative dopamine releasing effect and have pragmatically the same abuse liability. However, cannabis use often continues when people are prescribed opioids. So what about when you have a cannabinoid and opioid on board at the same time? And we still don't know exactly how this might affect the dopamine release, but we do know that using cannabinoids and opioids together does reduce actually the amount of opioid necessary to alleviate pain. And finally, cannabis has been shown to be able to treat various symptoms of opioid withdrawal, such as chronic pain or anxiety. So ultimately, it's a mixed bag. There are potential risk factors with cannabis use and later opioid use, but there is potential for cannabis to be used as a tool that we can leverage to make more effective, smarter decisions about when to prescribe opioids and how much.